Hello everyone, welcome to my channel Capsa. Today we will explore how bacteria share genetic material. Normally bacteria reproduce by binary fission, making the identical copies of the bacteria. That gives them numbers but not much variation. To adapt and survive, they use something called horizontal gene transfer. Horizontal gene transfer means DNA moves between bacteria, not from parent to offspring. It happens in three main ways. Conjugation is when two cells connect through a pilus and exchange DNA. Transformation is when a cell picks up nacked DNA fragments from the environment. Transduction is when viruses that infect bacteria, called bacteriophages, carry DNA from one cell into another. These processes are powerful because they give bacteria new abilities like antibiotic resistance or toxin production, making them stronger and harder to fight. Now let's talk about conjugation. So conjugation is often called bacterial mating, but it's not sexual reproduction. Instead, it's the direct transfer of DNA from one bacterial cell to another. Here is how it works. A donor cell called as F plus carries a special piece of DNA called the F plasmid. This plasmid has the genes to form a thin bridge like structure called a pilus. The pilus reaches out and connects to the recipient cell known as F minus, which does not have the plasmid. Once the connection is made, a single strand of plasmid DNA moves across into the recipient. Both cells then rebuild the missing strand. So in the end, each cell has a complete copy of the plasmid. What started as one F plus and one F minus now becomes two F plus cells. Sometimes the plasmid can even integrate into the donor's chromosome. When that happens, large sections of chromosomal DNA can be transferred to. These donors are called as HFR cells, which stands for high frequency recombination. In other cases, the plasmid carry along extra bits of chromosomal DNA, creating what's called as F prime plasmid. The real importance of conjugation is that it spreads useful traits very quickly. For example, antibiotic resistance genes can pass from one bacterium to another, even across different species. That's why conjugation is a major concern in medicine. Now let's move on to the transformation. Transformation is the process where a bacterium picks up free DNA directly from its surroundings. This usually happens when other bacteria die and break apart, releasing their genetic material into the environment. If a living bacterium is in what we call a competent state, it can take up this snacked DNA. Once inside, the DNA can join with the cell's own chromosome through recombination process. And if it carries useful traits, the bacterium gains new abilities. A classic example of this is Griffith's experiment with Streptococcus pneumoniae. He showed that harmless bacteria could become deadly after taking up DNA from heat-killed virulent strains. This simple experiment proved that DNA is the material of inheritance. In modern science, transformation has become one of the most powerful tools in biotechnology. For example, scientists routinely use artificial transformation to insert recombinant plasmids into the E. coli to produce insulin and other important proteins. Finally, let's talk about transduction. Transduction is the transfer of DNA from one bacterium to another with the help of viruses that infect the bacteria called the bacteriophages. Here is how it works. When a bacteriophage infects a bacterial cell, it uses the host machinery to make new virus particles. Sometimes by mistake, pieces of bacterial DNA get packaged inside the virus instead of viral DNA. When that defective virus goes on to infect another bacterium, it injects bacterial DNA rather than viral genes. The recipient cell can then incorporate this DNA into its own genome. This is called generalized transduction because any random piece of bacterial DNA might be transferred. There is also specialized transduction. In this case, the phage DNA first integrates into the host chromosome and lies dormant. When it later exits itself, it sometimes carry along neighboring bacterial genes. These specific genes are then moved into the next host cell during infection. Transduction is important because it can spread dangerous traits like toxin genes. For example, the diphtheria toxin and cholera toxin genes were acquired by bacteria through the transduction process. Now that we have gone through conjugation, transformation, transduction one by one, let's take a step back and compare them side by side. In conjugation, the DNA comes directly from a living donor bacterium. It requires physical contact between cells, usually through Pilus, and it's often plasmid DNA that is transferred. This makes conjugation the fastest way for bacteria to share resistance genes. In transformation, the DNA source is very different. Here, the bacterium takes up naked DNA fragments from the environment, usually from the dead bacteria. No cell to cell contact is needed, but the recipient must be in a competent state to accept that DNA. Transformation plays a big role in genetic engineering and in nature's way of recycling genetic material. Transduction, on the other hand, uses viruses as the delivery system. The source of DNA is another bacterium, but it's carried accidentally by a bacteriophage. Unlike conjugation, no physical contact between bacteria is needed. Instead, the virus acts as a messenger. Depending on whether it's generalized or specialized, the phage can transfer random DNA or specific genes located near its integration site. So, to wrap up, let's remember what we have learned today. Bacteria don't just rely on binary fission to make identical copies of themselves. They also have powerful ways of sharing DNA called horizontal gene transfer. Conjugation is like a handshake between two bacteria using a pilus to pass plasmids directly from one cell to another. Transformation is nature's recycling program where living bacteria take up naked DNA from dead cells and add it to their own. And transduction is when viruses, called bacteriophages, act as accidental messenger carrying DNA from one bacterium to another. Even though the methods are different, the result is the same. Bacteria gain new traits that help them adapt, survive, and sometimes become more dangerous. This is why antibiotic resistance, toxin production, and new metabolic abilities spreads so quickly in bacterial populations. So the key takeaway is this: bacteria may be tiny, but their ability to share DNA makes them incredibly powerful survivors. And understanding this process is not only important in microbiology but also in medicine, biotechnology, and public health. Thank you for listening carefully. If this lecture helped you understand bacterial gene transfer more clearly, please remember to like, share, and subscribe for more. Thank you.